Hey everyone, we're going to continue on our discussion about making ATP. We've already looked at cell respiration or oxidative phosphorylation as one way to make ATP, but did you know there's actually a couple other ways to make small amounts of ATP? It depends on what kind of organism you are, but there's two other types of ways that are anaerobic. That means without the presence of oxygen. Obviously, cell respiration or oxidative phosphorylation is when there is oxygen present, or aerobic respiration, but sometimes oxygen is not present. Therefore, the organism must do anaerobic respiration. But first, let's do a little review. First, let's take a look at the equation. This is with cell respiration when oxygen is present, using glucose as our model. Again, you can use other foods as fuel to do this, but glucose is our model here. So taking glucose and oxygen, which is then sent to the mitochondria, tearing it down again and again and again in many different reactions until you have carbon dioxide, which is a waste that you breathe out, water, and then of course energy in the form of ATP. Sometimes, if really good, up to 38 molecules per molecule of glucose. There's a lot of reactions though to get to that point, from starting with glycolysis in the cytoplasm of the cell, where glucose is initially broken down into pyruvate by a series of catalyzed reactions. And then that product of pyruvate being sent to the mitochondria, where it's sent into the citric acid cycle, and you continue to tear it down. All the while, what are you really getting from your food, from the fuel? You're getting electrons, which get carried by the hydrogens. And our electron carriers are NAD, which is in glycolysis. And then in the citric acid cycle, you have NAD and FAD. They become reduced by gaining the hydrogens, which also carries the electrons, and they both keep going and deliver those hydrogens with their precious electrons to the final step in the inner membrane of the mitochondria, where oxidative phosphorylation takes place in the form of an electron transport chain and chemiosmosis. All of this must be done in the presence of oxygen in the mitochondria in order to get that large amount of ATP. Glycolysis, by the way, is in the cytoplasm and can take place whether or not there's oxygen, but citric acid cycle and the electron transport chain must have oxygen present. Remember, oxygen is that electronegative molecule that pulls the electrons towards it. Hence the name oxidative phosphorylation. And don't forget our metabolic pathway here. Here we're in the mitochondria, and we have our series of enzymes, and we can see NAD is delivered here. NAD delivers its H, and FADH2 delivers its H's here for them, and then that frees them up to go back and go get more. So it's like you have to keep grabbing the hydrogens away from these molecules so that they are freed up to go do more, to keep this whole process running. If you don't take the H's away from them, then they just hold on to those H's and everything gets backed up and stops. So if there's not enough oxygen present, that could happen. Such as if you're out of shape and you're running for the first time. Maybe you've been sitting around all summer, you get back to school, and it's time to start Fayette again, and you're supposed to run the mile, and you haven't ran for a long time, you don't have a very good blood supply to your cells anymore. And so you could actually deplete the amount of oxygen at your mitochondria. So if that happens, you're going to have to use another method to make ATP. It will not be a very effective method as oxidative phosphorylation is, but there is another method to use. But we'll get to that in a second. Again, let's review our electron transport chain. As NADH and FADH2 give up their H's, the proton of the H, those are all pumped up here up to the top, and the electron, the H, is moved along these enzymes. Moving those electrons along these enzymes helps those enzymes pump the H's up to where they need to go. Meanwhile, oxygen is over here pulling those electrons towards it because it's electronegative. Oh, there's the diagram finished. NADH giving up its H, FADH2 giving up its, the electrons pumping. These are actually quite a few enzymes here, but they call them enzyme complexes. And then on the very end is our wonderful ATP synthase, the world's smallest turbine. So the whole idea, remember, of cell respiration is to gather up as many H's as possible from the food you eat and carry them to the mitochondria where they can go through electron transport, where the electrons can be pumped one way and the protons pumped another way. So you get an area, these little white circles, those are all H's. And those H's are what will be the energy to make this turbine spin. They can only go down, they want to go down their concentration gradient, that's called chemiosmosis, from where there's more of them to where there's less, plus there's electrons down here, so they're attracted to that too. So they'll travel down this little hole here of ATP synthase, they'll make it spin like a turbine, and when that enzyme is spinning, that's what gives this enzyme energy to put the P onto ADP to make ATP, which is what's supposed to be in this little star here. So again, that's the whole idea of cell respiration. If you're looking for the big picture, you're getting something to fuel the spinning of this enzyme to make ATP. And what fuels the spinning? It's the H's. You have to gather enough H's to make it spin so it can make ATP. And you can make a lot of them if there is oxygen present. But alas, sometimes oxygen is not present. If oxygen is not present, we can still make a small amount of ATP in the cytoplasm, and it would be called anaerobic because it would be in without the presence of oxygen. If oxygen is present, it's called aerobic. Without oxygen, it's anaerobic. And depending on which organism you are, you can do that. If you are a bacteria or a prokaryote, you can do alcohol fermentation. So that's one method of anaerobic respiration, alcohol fermentation. If you're an organism like you or a mammal, you can do lactic acid fermentation. That's the other method of anaerobic respiration. So let's take a look at these as to what they are. First, though, let's review this. 90% of the ATP that you make in your body is through oxidative phosphorylation through the electron transport chain, which is a great method because it makes so much. However, about 10% is generated during methods like glycolysis in a process called substrate level phosphorylation. If you looked at the diagrams of glycolysis, there's about 10 reactions or so, and some of those reactions are substrate level phosphorylation, where an enzyme grabs onto a substrate which is one of those intermediates of glycolysis, grabs onto a substrate that has a P available, grabs onto an ADP, and then gives that ADP a P, therefore phosphorylating ADP into ATP. So then you'll have one of those products of glycolysis and you'll have ATP. This does generate a very small amount, just a couple ATP. It's kind of inefficient, not like the electron transport chain, but it is capable of generating some when you really, really need it in a pinch. So first, let's look at what we do. So the whole idea to keep respiration going, you have to keep your electron carriers going. You have to keep freeing them up. You can't just stop them. You have to take their H's away from them to free them up to go gather more H's so that you can keep all of the reactions going. So that's the whole idea. And when you run out of oxygen in the mitochondria, then everything would stop, would get backed up, and your electron carriers like NADH would just sit there with their H's, which would then stop all the other reactions behind it. And that is not what you want. So in circumstances when you are lacking oxygen, you can go into lactic acid fermentation. It kind of sounds bad because in a way it kind of is. It's inefficient and you don't make very much ATP. And because of the products that you make from it, you've probably heard of lactic acid before, you're going to be in some pain for a few days when you have to do this. So the idea is that we can still do glycolysis in the cytoplasm. It does not require oxygen, so we can still do it. However, we have to keep freeing up our NADs in order to keep the glycolysis reactions going. So that means NADH has to have some molecule to give its H to so that it can become NAD again and then get back into those glycolysis reactions and keep receiving H's. So literally, the product of glycolysis, pyruvate, instead of going on to the Krebs cycle in the mitochondria, actually becomes the electron or the H acceptor. So this is pyruvate here, and pyruvate will accept this H off of NADH, and if you look at the product, you can see exactly where it goes. It goes right here on this O. There it is there. So literally, pyruvate and the molecule lactic acid, actually called lactate, really are not that different. Lactic acid just has an extra H on it. So pyruvate accepts the H, so NAD can get back in there and keep glycolysis going. But pyruvate becomes lactic acid. And this will generate only 2 ATP, which is not a lot. All right, you probably have heard of lactic acid. It has that bad stigma that it causes pain in your muscles as you try to get rid of it in the next few days. And that might be so, but there's newer research out there that it's not really just the lactic acid causing you the pain. It's also, they're linking it that there may be more potassium ions involved and the breakdown of your muscles for doing new exercises that you weren't used to are also culprits in causing that pain of the buildup of lactic acid. The good news is that lactic acid then, eventually in the next few days, will be sent to the liver, and the liver will convert it back to pyruvate. And because everything has settled down now, you can now send that pyruvate that you make from that to the Krebs cycle, which then it can be involved in oxidative phosphorylation or the electron transport chain. All right, so again, the idea is you have to keep freeing up your NADs so that they can accept H's and keep the reactions of glycolysis going. So pyruvate becomes the electron or the H acceptor, and then becomes lactic acid. And you generate just a couple ATP during lactic acid fermentation. Well, that's you. Well, there's other organisms out there too. How do prokaryotes do it? Well, remember, prokaryotes don't even have mitochondria. And so this is happening for them. They can do alcohol fermentation. So this looks a lot like what we did. We've got glycolysis happening. We're generating a couple ATP. They're making pyruvate. However, with these prokaryotes, this top CO2 comes off and becomes carbon dioxide. And then this molecule receives the H. You can see exactly where it gets put right here. This O becomes OH. And well, then this becomes an alcohol called ethanol. And so that's why it's called alcohol fermentation. And this process is often done in making different types of alcohol. If you feed your microorganisms glucose, they will indeed make alcohol through this alcohol fermentation process. And then you could drink it. It's a good thing that when we're doing lactic acid fermentation, we don't make alcohol. We'd probably get drunk or something like that. I don't know. But this alcohol fermentation is what microorganisms do. Again, the same idea, keeping NAD moving, giving it a molecule to give its H's to, so that it can go back in, get back into the fight, and keep taking H's from glycolysis to keep those reactions going and keep making at least a little ATP. There's some different types of organisms. The obligate anaerobes can only do fermentation. In fact, if you put them in the presence of oxygen, it will kill them. So they permanently only do this. Then there's facultative anaerobes. They can actually switch back and forth and do both. Now, we're so used to talking about breathing oxygen because that's what we do. And that oxygen goes to the mitochondria and is electronegative and pulls those electrons towards it. Well, it turns out that some organisms live in a place where there is no oxygen, such as deep sea bacteria. There's no oxygen down there. So what do they use as their electronegative molecule to help pull electrons towards electron transport? Well, it turns out that SO4, a type of sulfate, is also electronegative, not as much as oxygen, but still electronegative enough to make it happen. And at the very end, if you can imagine, when we have our oxygen waiting there, it combines with the leftovers, which are the hydrogen protons and electrons, or oxygen combines with the hydrogen to make water. Well, theirs is a little different. Their sulfate combines with the hydrogens coming off of it to make H2S, which is hydrogen sulfide gas, which is what makes, like rotten eggs, stink. So back down into these deep sea areas, these deep sea vents where these bacteria live, there is no oxygen. I can imagine how stinky it is from their process of making ATP, as they're making so much H2S down there. All right, so in summary, fermentation, anaerobic respiration, and aerobic respiration are three alternative cellular pathways to produce ATP by harvesting the chemical energy of food. All three of them use glycolysis to oxidize glucose or take its H's or electrons away from it, and then make pyruvate, and can net about 2 ATP by substrate level phosphorylation. All three of those pathways use NAD as the oxidizing agent, which accepts the electrons or H's from the food during glycolysis. A big difference, though, is how you turn NADH back to your NAD, which is required to keep glycolysis going. In fermentation, the final electron acceptor is an organic molecule like pyruvate, such as in lactic acid fermentation or alcohol fermentation. But in cell respiration and electron transport, electrons carried by NADH are transferred to an electron transport chain, which then can regenerate your NAD, which is required to keep glycolysis and all those reactions moving forward. Another major difference is how much ATP you actually make. You make a lot using an electron transport chain through oxidative phosphorylation, but in fermentation or anaerobic respiration, you only make a few molecules of ATP at a time. Then finally, speaking of glycolysis, the role of glycolysis in both fermentation and cellular respiration has an evolutionary basis. Ancient prokaryotes are thought to have used glycolysis to make ATP long before oxygen was even present in Earth's atmosphere. The oldest known fossils of bacteria date back to about 3.5 billion years ago, but significant amounts of oxygen probably didn't begin to accumulate in our atmosphere on Earth till about 2.7 billion years ago. Cyanobacteria produced this O2 as a byproduct of photosynthesis. Therefore, early prokaryotes may have generated ATP exclusively from glycolysis at first. The fact that glycolysis is today the most widespread metabolic pathway among Earth's organisms suggests it evolved very early in the history of life. The fact that glycolysis actually occurs in just the cytosol or cytoplasm of the cell also lends support to that idea. And that pathway doesn't even require any membrane-enclosed organelles like the mitochondria would be in a eukaryotic cell. And the eukaryotic cell evolved approximately 1 billion years after the first prokaryotic cell. So glycolysis is kind of like a metabolic heirloom from early cells that continues to function in fermentation and as our first stage in the breakdown of organic molecules by cell respiration today.